All right, back to topic 10. So last time we talked a little bit about the history of antibiotic discovery, and we also talked a little bit about selective toxicity. So selective toxicity, if you remember, is this whole idea of the magic bullet, the whole idea that you are going to find a chemical or a substance that is able to kill the microbe and hopefully not have any effects, or at least minimal effects on the host. And I finished off in this slide, basically where I had said that in terms of the number of drugs we have the most antibacterial drugs. And that's because bacteria are very unique compared to us. They are prokaryotes, we are eukaryotes. That gives them a lot of unique cellular structures that are actually targets for drug action. Fungi, protozoa, and parasites are also eukaryotes like us, so there are less differences. And viruses don't have cells. In fact, they're using our machinery, so it makes it a little bit harder to find effective antiviral drugs. So these other drugs, like I said, we'll get into them next day. Today, mostly we'll be talking about antibacterial drugs. So antibacterial drugs. Like I said, the biggest strategy here is to find something that is unique to the organism, to the pathogen, that we can disrupt and not disrupt something similar in the host, which is the human. And the biggest class, actually representing several classes of these drugs, has to do with cell wall synthesis, so peptidoglycan metabolism. There's a few other categories here. We're actually going to be covering 5 categories of antibacterial drugs. Like I said, the biggest one is, of course, the drugs that are affecting the bacterial cell wall, and the most important drug we have on here are the penicillins. So most of these names you'll have to know to some degree, not all of them necessarily. I borrowed this from something else, so not all the drugs we're going to cover, but you should know one from each category at a minimum for the midterm. So let's talk about penicillins. Like I said, this is the biggest group of drugs, the most important group of drugs historically, and even to this day, probably the most important group of antibiotic drugs. And these inhibit peptidoglycan biosynthesis. So let's talk a little bit about that and how they work. So there's a whole bunch of enzymes involved in cell wall construction in bacteria. And these enzymes go by a whole bunch of different names. A bunch of them are known as penicillin binding proteins. So penicillin binding proteins are actually enzymes, and they were discovered long ago, and we didn't know what they did, other than that they abound to penicillin. So that's how they got their name. It doesn't really, not really the best name in terms of the enzymatic activity, but this is what they do. And you can see they form a complex, and what they do is they build the new peptidoglycan. And so the question is, what does penicillin do? Penicillin goes in there and disrupts some of the enzymes. And what that does is it leaves holes in the cell wall. And of course, holes in the cell wall leads to poor structural integrity, and the cells literally burst open when they're growing. And so you can see that other term I have, beta-lactam. That's another term for penicillins. And I'll show you some pictures of that in a minute. So here's just another little graphic trying to show you what's going on, right? We have our cell, and a normal growing cell, when it grows, it has to make space for new materials. And so this is actually a pretty good analogy. If you think about if you're doing a renovation at your house and you have to do something with your wall, often involves actually tearing out drywall and those kind of things. If you want to build a new wall or expand a room or whatever, same idea with bacteria is they actually have to make spaces for new materials. And in the growing cell, the new material is put in, and then eventually it divides, and that's kind of the normal situation here. When penicillin is involved, the same thing happens. It starts to stretch out, it starts to make space for new materials. The drug penicillin is like throwing a wrench in the machinery, and the whole thing kind of has some issues, and it collapses. And so there's my cell. I think I actually have a video here. Let me show you here. But on the next slide, there we go. Yeah, there's a little video here. I'll play this for you. And This is better than the one I had. The previous one I had was cool because it had sound effects. Unfortunately, this one does not, but I'll play this for you. And what you're going to see is growing E. coli cells treated with ampicillin, which is a type of penicillin. And you'll see they're going to grow, 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 and then they're just sort of going to burst, sort of like putting too much air into a balloon. So here we go. They're stretching out, and watch each of these cells is going to one by one kind of just burst open. So, like I said, another analogy is thinking about like blowing up a balloon and eventually it explodes. That one in the top right, I have no idea what's going on there, but it's pretty cool. Anyway, that's kind of how penicillin works. This was actually the subject of my PhD thesis in a sense. And so I know quite a bit about how penicillin works and the action and all that. Let me show you some pictures here of some penicillins. Like I said, these are also called beta-lactams. Beta-lactams is it's kind of the term given to them by chemists. That little square ring that you see in the middle, you can see I have them highlighted in yellow with the nitrogen and the double bonded oxygen sticking off it. That's actually a beta-lactam ring. So this whole group of enzymes is called beta-lactams. Or penicillins. I usually refer them to them as penicillins because people are familiar with that word and it's accurate. And you can see there's a whole bunch of them. In fact, there's hundreds of these drugs. Penicillin G, the one there on the left. So this one here was the original penicillin, and you can see that it is injectable. That means that was the route people had to take it. And so that's not necessarily the most desirable drug. And I mentioned it wasn't very stable, and it's only really active against gram-positives. And so it didn't take long before we started to discover and synthesize other penicillins. So you can see penicillin V here, oral and acid resistant. Acid resistant, of course, is huge because that means we can put it in a pill. People can get a prescription and take it orally, and it's not a big deal. Penicillin B is actually still prescribed pretty commonly today. Back when I was a university student, I had pneumonia, believe it or not, and this is what they prescribed to me. And so the interesting thing was my student drug plan, of course, covered the drug, but not the dispensing fee. And I looked, and the dispensing fee was $16. So I had to pay $16 for my prescription, and the drug was $4.25. So this has been around a long time. It's super cheap and quite effective. There are other drugs here you can see on this list. Ampicillin used to be really commonly prescribed because it's acid resistant and a little bit more broad spectrum, meaning it's effective against gram negatives and gram positives. And we'll talk about what broad spectrum means in a few minutes here. And a bunch of other drugs on here that have various uses, a lot of them being broad spectrum or a little bit more powerful or a little bit more active against some organisms. A really important one is methicillin. And we talk about methicillin a lot in bacteriology circles because it used to be considered the drug of last resort, meaning if an organism was allergic or resistant to everything else, they would maybe still be killed by methicillin. So we'll come back to methicillin a couple of times here, particularly when we're talking about MRSA. Okay, so that's kind of it for now about penicillin. Like I said, I'll be kind of mentioning it on and off. I know I've been mentioning on and off all semester. So if you're going to learn any one drug in this lecture, know about penicillin. Obviously, there's way more we could say about it, but this is about the level that we need for this class. I do want to introduce some other drugs today. So there are other drugs that inhibit bacterial cell wall synthesis, different classes of drugs with different mechanisms. I'm not going to cover them all, but the one that I do want to talk about is this one here, isoniazid. And you can see that this one here is not affecting peptidoglycan synthesis, but something else found in the cell walls of certain types of bacteria, the mycolic acid. So if you remember, we were talking about mycolic acid as being a unique feature found in the mycobacteria, so such as Mycobacterium tuberculosis, and that it was kind of a waxy substance, and it made it really hard for these things to be gram stained, and actually protects them somewhat against immune cells. And so these are tough little organisms. And good news is we have found a drug that will actually affect the mycolic acid synthesis, and therefore is a relatively effective antibiotic against Mycobacterium. And there's the structure of it. There's the Mycobacterium tuberculosis bacilli there in the micrograph. And so this is one worth knowing as well. We're going to come back and talk about a little bit when we talk about tuberculosis in topic, I think it's topic 14, we're going to talk about tuberculosis in a little bit more detail. There's our mycolic acids. I think I showed you this slide before, just mentioning that they're part of the cell wall structure, and they're protective, and when you remove that protective layer, the cells are vulnerable. All right, so moving on, like I said, it's kind of like a little bit of a big list here, but you're going to see that that common theme of selective toxicity, in that we're targeting things that are unique to the bacteria. So another group here of drugs are the polymyxins, and these target gram-negative bacteria. So if you remember gram-negative bacteria, they have that extra layer, and that extra layer of membrane involves these LPSs. So LPS stands for lipopolysaccharides. And that is something that is unique to the bacteria. So this makes it a good target for antibacterial drugs. So there's polymyxin. I just love some of these antibiotics. If you like chemistry, just looking at some of these molecules, it's just mind-boggling that that these organisms are going to synthesize these crazy things. That's what polymyxin B looks like, and you can see there's our gram-negative bacteria in the outer membrane, which is made of LPSs, and there goes the polymyxin. So that disrupts the outer membrane and causes permeability and holes and those kind of things, and it kills the bacteria. Now these ones here, I don't know a lot about the polymyxins. I think there's something that can cause side effects in humans because, of course, we do have membranes. So they're not typically used as oral drugs. They're more surface drugs. And so what I mean by that, I mean like creams and stuff that you're going to put on wounds. And usually at this point, somebody at least has kind of thought polymyxin. I know something that my mom uses with the word poly in it, polysporin. And so this is a surface antibiotic ointment. And some people, if your mom was like my mom, she was a little bit crazy over this stuff. Any little scratch that we put a Band-Aid on when I was a kid, it's like, let's get out the polysporin. And then, you know, that was kind of the part of the treatment. You can see there's a few different formulations of polysporin. This one here in particular actually has 2 antibiotics in it. It has polymyxin B and bactracin, I think is how we say it. And there's, like I said, there's other formulations of this. I saw this one on the shelf as well. This one has 3 different antibiotics in it, so I don't know whether that one is supposed to be better. And I was looking on the shelf in the pharmacy, and there's different, like I said, different formulations of polysporin, some with 2, some with 3. I don't know why they don't just have one formulation. Probably has something to do with shelf space. You know, the more shelf space you get, the more likely you are to sell your product. Okay, so Group A, cell wall synthesis. Group B is membrane damage, more for topical antibiotics. So Group C is maybe one of the other big important groups of where there are lots of drugs. And these are antibiotics that affect protein synthesis. So there's a whole bunch of drugs that fall into this category. We're not going to learn them all. I'm going to introduce you to 3 of them. And if you remember what protein synthesis is like, it's also called translation. And so maybe I'll write that down, translation. So translation, of course, is a process that happens at the ribosome, and at the ribosome, the messenger RNA is bound, and the ribosome reads the codons on the messenger RNA, and it's going to assemble the protein chain. So it puts the amino acids together. And so the ribosomes I keep going back to this slide because prokaryotic ribosomes are unique. They're different than eukaryotic ribosomes. They don't look too much different in my picture, but if you take a look at the details, the prokaryotic ribosomes are smaller. They're 70S versus 80S for the eukaryotic ribosomes. If you look at the components, they're made of RNAs and proteins, and they're different, or at least they're different enough that these are legitimate drug targets. And so if we can disrupt these ribosomes, we can disrupt growing bacterial cells. So let's take a look at some of these. One of the most important drugs on this list is erythromycin. And erythromycin, by the way, again, another really cool chemical compound. This is a really important drug. And this is the drug that they will often prescribe to people if you are allergic to penicillin. So if you're allergic to penicillin, they don't want to prescribe you any of the penicillin drugs, any of the beta-lactams, because they're concerned you might have a reaction to that. So what they have to do is they have to look for a drug outside of that class. And this is kind of the number one choice, the erythromycin. If you're allergic to penicillin, they're going to prescribe you erythromycin. Erythromycin is pretty common as well for bladder infections, which in many cases is caused by certain strains of E. coli. And I'm not sure why it's the drug of choice for bladder infections, or at least it used to be. I don't know whether that's still the case, but it is. So you can see what it's doing is it's actually binding the large ribosomal subunit, so the 50S subunit. So that's why I have the arrow up to the top. That's details you don't need to know. The main thing to know is that it is interfering with ribosome function and blocking protein synthesis. Like I said, there's a whole bunch of these drugs that bind ribosomes in different ways and lots of interesting chemical structures. Here's another one, tetracycline, also a really commonly prescribed antibiotic. And you can see it is binding the other part of the ribosome. It's binding this small subunit. And tetracycline tends to be a very potent antibiotic. And sometimes it's prescribed after something doesn't work. Sometimes it's prescribed for, it's a very effective broad spectrum, meaning it can kill a variety of different organisms. And there are some organisms that might be naturally resistant to other antibiotics, and tetracycline is still effective. And so it's really commonly prescribed, although it's a very powerful drug. And there are some issues with tetracycline that I'll mention here in a second. I think I have a picture coming up here. Let's see here. There's a picture. Yeah. So one thing about tetracycline is it is known to bind to growing bone. And it's also the compound itself is yellow. I worked with tetracycline, and it's a yellow powder. And we try to avoid prescribing these to children because, of course, they have growing bones. And one of the biggest issues, of course, is cosmetic, in that it can cause yellowing of teeth, particularly because children have growing teeth. My understanding is it can also weaken growing bones a little bit too, but I'm not 100% sure about that. I'd have to look that up. I know this is one of the major things, and if you Google like tetracycline teeth or something like that, there's all sorts of dentists out there that have procedures that they want to help you with this kind of thing. So those are the two to really know, erythromycin and tetracycline. I wanted to mention one other drug that is just mostly important historically, streptomycin. Streptomycin was actually the very first drug to treat tuberculosis. So this drug was super important historically, and I think I have a note there on there in that we don't really use it anymore because it is also known to cause ear damage and can cause hearing loss. And this is the case with a lot of these antibiotics. Once we start to get out of the beta-lactam range, is that humans do have ribosomes. And so some of these drugs do have side effects that depending on the drug, particularly with prolonged use. So tuberculosis treatment can range anywhere from 6 months to 12 months. So you can imagine taking a drug for that long, and even if it has a tiny amount of toxicity to the human, when you're looking at long-term use, it can cause issues. And in the case of streptomycin, it was ear damage, hearing damage. So it's worth it to mention, like for historical reasons. I won't ask you about streptomycin on the exam, but I just wanted to mention that because well known for its side effects. Okay, moving right along. So I'm sorry, there's lots of weird words for these drugs, and it's just kind of the nature of the beast. Wait till you take pharmacology, it gets really weird. Some of them have functional names, some of them it's just random letters put together. And um, maybe I'll talk a little bit about nomenclature of these things at some point if I get a chance. So group D are drugs that inhibit DNA synthesis. So you can see this, this word here, fluoro. So these things are going to have fluorine in them. And there's actually ciprofloxacin, which is kind of affectionately called Cipro. This is a relatively new antibiotic. It's only been out for about 20 years. So I think it's had a lot of prescriptive use in the last 20 years. So what's going on with DNA replication in bacteria? So you may remember that DNA replication is where you have one strand of DNA. So that's shown there on the right. And it's going to make two daughter strands of DNA. And it involves this enzyme here. So DNA polymerase. So it turns out DNA replication actually has a bunch of enzymes and proteins that are involved in the whole process. It's a pretty complicated process. And so one of the proteins that are involved in this whole thing is called topoisomerase. There's another one that's kind of similar called the DNA gyrase. And so those enzymes, what they do is they untangle the DNA. You could imagine DNA as a helix. And it's kind of like if you had a rope, or let's say you had braided hair, you're not just going to tug at it because it's just going to form a tighter knot, right? And so we actually have an enzyme in there. And it turns out that the bacterial enzymes are unique enough from the human enzymes. So these are, you could call them, probably a better name for these enzymes would be untangolase or something like that. But that's what they do. They're untangling enzymes. And so these fluoroquinolones, they basically get in there and they're blocking these enzymes. And so the bacteria cannot replicate their DNA. They can't grow. And that's how the drug works. So like I said, these ones here are usually considered relatively potent. They're sort of a newer class, particularly the Cipro. And in some cases, they're being used to treat organisms that are resistant to other things, or at least other classes of drugs. But these things are changing all the time, the standard usage, because of course, we have better practices of care, and sometimes we're dealing with things like superbugs, which we'll talk about in a couple of minutes. All right, so the last class of drugs we've mentioned already, and depending on the textbook, sometimes they just say drugs that inhibit metabolism, because there are a lot of metabolic pathways in bacteria that are unique, right? And kind of the main drugs for this are these sulfonamides. So if you remember the sulfonamides, I talked about them last day. I mentioned that these are what was the drug? Prontosil was one of the historical drugs that was discovered, and what was it being used to treat? Streptococcal infections, right? And so basically what we're looking at is another pathway to the organisms. So in this case, this is the pathway that's used to make folic acid. So you may know a little bit about folic acid. Folic acid is one of the B vitamins, and as humans, we have to get it in our diet. So it's one of those things that if you're pregnant, you take the prenatal vitamins, and it has extra folic acid in it, and those kind of things, right? So it's essential for humans. But the difference is that humans, we can't synthesize it. We have to get it in our diet. Whereas bacteria, they don't get it in their diet. They actually synthesize it. So if we can prevent them from making it, then we can kill them. And that's exactly what we do. So you can see this pathway is showing that it starts off with this compound called PABA. There's a bunch of enzymes in there. Eventually it makes folic acid, which eventually is used to make nucleotides. So again, you can't make nucleotides and you die. So there's our sulfonamides. You can see it kind of looks like that compound PABA, and it binds these enzymes, and that's how it kills them. So these are not as commonly used as others. They tend to be considered not as potent. but they still have their uses in medicine, and just not as common as the other ones, because, like I said, in terms of potency, these tend to be a lot less effective. Okay, so you need to know these ones for the exam. Okay, what am I going to have on the midterm? I think last year I had a matching question, so maybe you didn't need to know all of them, but you should if you're looking to learn a minimum list, it's this list here, and the macrolides, of course, that one uses erythromycin. Macrolides is just the class of those drugs. Okay, so I know there are a few others mentioned in there, but those are the ones that you need to know for the midterm. And if you're really looking to focus, you definitely want to know penicillin. Okay. All right, so my goal was to not make the list super long. My goal was to kind of just give you a little bit of context for each of these categories of important antibacterial drugs. And like I said, penicillin being the most useful of them. And you can see that whole concept of selective toxicity, something that is there in the bacteria, is unique to the bacteria, and therefore makes it a target for our drugs. So let's move on. I want to talk about a few other things regarding antibiotics. And I know I mentioned this term already, this idea of broad spectrum antibiotics. And so broad spectrum means it can kill a variety of different bacteria. So something that, let's say, kills both gram-positive and gram-negative organisms. Narrow spectrum antibiotics are a little bit more limited. So you may remember that penicillin G, I should say G beside it, is something that's really mostly effective against gram-positives, but not gram-negatives. I had mentioned that the polymyxin is more effective against gram-negatives than gram-positives. And so both of these types of drugs have their uses. And so, I know I have in the notes here, I didn't fill it in. Usually I do this on the board or something like that. But I want to talk about kind of some of the advantages and disadvantages of using broad spectrum antibiotics. So what are the advantages of using broad spectrum antibiotics? Well, they're effective against a whole variety of organisms. Somebody has a skin infection or pneumonia or whatever, you don't know what bacterium is causing it. You can prescribe, something relatively potent, and you don't need to wait for culture results or things like that. So what's the disadvantage? The disadvantage is that we have a lot of microorganisms associated with us that are good organisms. And we're going to be talking about this after the midterm in terms of our normal microbial flora. And we don't necessarily want to do that, right? Let's say somebody has a skin infection and you treat them with broad spectrum antibiotics, right? And so this is going to wipe out and kill many bacteria on the body. And this can lead to, let's say, a yeast infection. So this is actually the case with a lot of women. They get ill for some reason, you get prescribed antibiotics. The antibacterial drugs will kill many bacteria, including many of the bacteria in the vagina. And this gives a chance for the yeast, because yeast is, of course, a fungi, which is a normal part of the vaginal flora, to basically flourish, and then can cause a yeast infection. And so, this is the case for many many secondary infections, and I'll probably mention a few more as we go along over this semester. So just some definitions for you, and I'm not really going to get into this, and I won't be asking you about it on the test, but it is mentioned in the textbook. Sometimes drugs are distinguished in terms of, are they bacteriostatic or bactericidal. So bacteriostatic means they don't kill the organisms. Bactericidal means that they do. And again, these things have their uses, and we're not going to get into this anymore. I won't mention it again, but like I said, these terms do pop up, so I wanted to just throw them out there for you. Okay, so I want to talk about a little bit how we can test the effectiveness of antibiotics. And I mentioned it's very similar to what we do with our disinfectants. We can do this disc diffusion assay. It's also known as the Kirby-Bauer assay. Those are some scientists, I believe, who kind of developed it. And it's really the same idea. So you see here, you've got these little discs. The one in the middle is yellow. So that one is tetracycline. And the one on the right here, GM, is gentamicin, chloramphenicol, probably that CF, ampicillin, the one at the top. K is kanamycin on the left, and penicillin B is the one in the bottom left corner. So you can see they all have little numbers beside them. That's probably the concentration, so micrograms per mil. And you're looking at that zone of inhibition. So the bigger the zone, the more effective the drug. So you can see penicillin B, whatever the drug this is not so effective at 300 micrograms per mil, whereas tetracycline is effective at 30 micrograms per mil and a much larger zone of inhibition. So this is a pretty common kind of thing that's done, like I said, with disinfectants and antibiotics. It's a quick test. Sometimes the numbers aren't perfect because it sort of depends a little bit on the technician, but it's a very quick test, very visual. You can see what's going on. The better test is something called the MIC, or minimal inhibitory concentration test. And I showed you this with the disinfectants. And really the whole idea is that you make a dilution series. So you're adding the drug to the test tubes and you're getting, in this case here, the concentration is increasing when you go from the left to the right. You can see the concentrations here. So that's 0.2 micrograms per mil, 0.4, 0.8, 1.6, 3.1, and so on. So it looks like they just doubled it as they went along. And in this case here, you can see that the minimal inhibitory concentration is 1.6 micrograms per mil. Anything lower than that concentration is not useful. So often these kind of tests are done to compare antibiotics. And in the case of therapies, you're looking at trying to understand what the effective dose is going to be to give the patient. And so sometimes these things are done behind the scenes when they're dealing with problematic cases of resistant organisms and so on. So one more test I want to show you, which is kind of cool. We've got the disc diffusion. That, like I said, this is quick and easy to do. This one here takes a little bit more pipetting, but gets you good results. So there's a company that has made these little sticks here. I can't remember the name of the company. I think it's BioMaru. And they've decided to combine the MIC and the disc diffusion all in one test. So what they've done is they've made these little sticks, I guess you call them, made out of filter paper. And embedded within the filter paper are different concentrations of the drug. And so you can kind of do all this test at once and at least get a rough idea of the MIC very quickly using the e-test. So these are pretty cool. And like I said, looking for quick results, you can get an estimate of the MIC very quickly and get those visual results. It involves way less pipetting, much, much quicker to do. So back to our timeline. I showed you this timeline here, and we were talking last day about antibiotic discovery. And I'd mentioned that many of the drugs we use were actually discovered in the 50s and 60s. And really, there was not as much research going on for quite a number of decades until the 2000s. And in fact, many of these drugs were actually discovered before, but not really pursued because we had hundreds of drugs. So what's been going on? Like, why have we not been doing this research? And why did we stop? And why are we picking it up now again? So the answer is resistance. Bacteria have become resistant to many of our traditional antibiotics. This is a bad thing. So why is this? Bacteria are living out there in the environment. They're living in the soil, they're living on skin, and they're not just dealing with humans. In fact, if you think about soil organisms, there are in a scoop of soil, probably 10,000 species. And they're all competing with each other for that biological niche. And if you remember, the penicillin is a mold, and the mold is killing the bacteria. And this is kind of the way of their life, is they're used to competing with one another and making drugs. So it's no surprise that that these things actually, that genes for resistance are actually out there. And it really didn't take long before these resistant organisms started to emerge in healthcare settings. Even in the 1940s, there was some evidence of this kind of thing happening very, very quickly. If you take a look at this one here, there are tons of graphs like this showing resistance in bacteria. This one is for Neisseria gonorrhea, looking at, it doesn't say which drug it is, but between the years of 1980 and 1990, basically going from almost 0% to almost 9%. And we see these trends all the time. You know, a new drug ends up on the market, and you know, they have about a 10-year, not necessarily life cycle, but you got about 10 years before you're starting to get some serious problems in many cases. And so this is something that you know, we're trying to deal with on a constant basis. And I'll give you some hints and ideas of what is being done on these things. But like I said, resistance is a serious problem. So I just want to talk a little bit about where and how the resistance works. Biologically, there's kind of three main mechanisms in terms of how resistance works. First mechanism is basically the organism destroys the drug somehow. So you can see in that case there we have an antibiotic. Let's say it's penicillin, and the organism has a gene called penicillinase, and the penicillinase can actually chew up penicillin and therefore make it useless. Sometimes the drug doesn't get destroyed, but it might get altered. So it's usually considered a different class because the drug may still be effective against other organisms, kind of thing. And so you could take penicillin and maybe add a functional group that blocks its ability to bind the penicillin binding proteins, for example. And then the last category are these pumps. They're called efflux pumps. Efflux basically means out, right? So it's pumping these things out. And it turns out that bacteria have all sorts of transport proteins in their membranes. And some of these can bind antibiotics and push them out. So it goes in the cell, they push it out, and the drug becomes less effective. Sorry, I missed your question. Somebody has a question here about, are there more side effects with narrow versus broad spectrum antibiotics? Probably more side effects with broad spectrum antibiotics. As I mentioned, you're looking at killing helpful microorganisms and those kind of things. And I mean, it really just depends on the drug too, right? Like I said, you know, the penicillin group of drugs tend to have less sort of side effects on human physiology. The more the side effects tend to be, you know, that you're changing the flora. So it's kind of a complicated answer on that. Hopefully that helps a little bit. So just to kind of finish this off here, I just want to show you that, you know, in terms of these mechanisms of resistance and relating this back to the drugs that we were just talking about, these types of resistant properties are found all over the place. Pretty much for every class of drug, there are multiple mechanisms of resistance, just depending on the actual type of organism. And so it's a pretty complicated and big story on this. So I want to talk about how these things get resistance. And I found this little cartoon. It's just wonderful. You can see it says here, it was on a shortcut through the hospital kitchens that Albert was first approached by a member of the antibiotic resistance. And so the shady character says, hey kid, want to be a superbug? Stick some of this into your genome. Even penicillin won't be able to harm you. So this is actually a pretty good lesson. There's a lot of stuff packed in here. And this is really much more important about the mechanisms of resistance is to how they have become resistant. And so let's take a look at this. There's actually kind of two main mechanisms. And we call them, in biology terms, we call them vertical and horizontal transmission. And I'll sort of clarify what that means in a moment. But one mechanism is really just selection, okay? So this isn't natural selection, by the way, because it's not happening out in nature. But it's a type of, you know, kind of related to that in terms of humans are creating a situation that is allowing selection. So take a look at this. You have a pool of bacterial organisms, and they grow and they reproduce by binary fission. And it turns out that there's always a few mutations in there with these organisms. And so, you know, if you think about the mutation rate, it's not very common. It's like 1 in a billion nucleotides or something like that. But if you think about what's going on, if you're growing a population of E. coli in a lab, I can have 1 billion cells overnight. So the chance of mutations is low, but because these things multiply in great numbers, the actual chance of it happening is frequent enough that sometimes you get small differences in the individual cells in the population. Now, once in a while, one of these mutations might be helpful to the organism. For example, it makes it a little less susceptible to a drug. So you can see this, this dark one here. It says this one is drug resistant, or at least a little drug resistant. So what's going to happen when this person takes the drugs is it's going to kill off these lighter colored ones, and this darker colored one is resistant, so it is able to live. And of course, since it lives, it can now multiply, and now you can have a drug-resistant population. So these changes are relatively incremental, and I'll show you a video in a second, though, of this whole thing happening in real life, and it's well documented, and it's kind of scary. Let me just take a look here. And so this is kind of why usually, if you've ever had antibiotics, usually what the doctor will tell you is he or she will say something like, and make sure you take all of the pills. So let's say the prescription is for 10 days. Make sure you take them all for 10 days. And typically what happens is you take the drugs and, maybe day 3 or 4, you're feeling a lot better, but they want you to take it all the way to the end. And the whole idea is that so that you have relatively high concentrations of the drug in your body, and that you're going to wipe out all of these ones that are maybe partially resistant. If you just take small doses of antibiotics, this kind of selection is a lot more likely to occur. And this is something that we're very concerned about, of course. So vertical transmission, you can see, basically means you're going from one generation to the next, right? So in humans, vertical transmission means that you might give your traits to your children, right? And so the other type of resistance mechanism is called horizontal transmission. And this means it's not coming from a previous generation, but it's coming from other sources. And this tends to be a lot more rapid, but also the frequency is small, but it does happen out there in nature. And there's a whole bunch of different mechanisms, kind of 3 that I have here that are worth mentioning. So we talked about bacterial conjugation. Do you remember way back in topic 3, we were talking about the sex pilus and that mechanism for organisms to exchange DNA. And this can be done between different species, which is kind of the scary thing about it. So you could have one organism that ends up in a hospital, and it transmit genes for resistance to other unrelated organisms. And sometimes it's multiple genes for multiple forms of resistance, which is really quite terrifying. There are other mechanisms. Another mechanism is called transformation. So some organisms have the ability to scoop up DNA from the environment. So gonorrhea is very good at this for some reason, and cells die all the time. Gonorrhea samples DNA. If it's a useful gene, it's going to keep it, right? So this is just, like I said, another mechanism, not relevant for all organisms, but some. And another mechanism is something called transduction. So viruses, of course, are instruments for injecting DNA into things. And once in a while, not very frequently, but it has been observed, genes get transferred between organisms via viruses. So the mechanisms are less important than understanding that DNA can come from multiple sources. And you can see there's our organisms, and they get transformed, and some of them change. And you can see I've given them evil-looking eyes, and some of them end up with multiple genes for drug resistance. So that one you can see has horns and a tail, it's a little extra nasty looking there. And these are the ones we're really scared about that have multiple forms of resistance, of course. So I told you I wanted to show you a video. It tells me it's not a valid video or cannot be embedded. So what I will do is load this up for you, and I'll let the video speak for itself. But let me just say, it's a good thing it's light out, and this is one of the most terrifying videos on the entire internet. Just in time for Halloween. So I'll load this up and we'll play it, and you'll see what is going on here. So just give me a moment here. I'm just going to get rid of the advertisement. and of course share my screen. Okay, where is it? There we go. All right, I'll let it speak for itself.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Okay, so like I said, absolutely terrifying what is going on there. I mean, there's a few caveats around that in terms of which antibiotic we're using. Some antibiotics, it's much easier to select and become resistant to, 1,000 times is a little bit extreme. It's not going to happen with most antibiotics, but the principle is there, right? There's a selective pressure, and the organisms have the ability to adapt, and it can be absolutely terrifying. So that's, like I said, why it's important for people to finish their prescription to make sure that they do have high concentrations of the drugs, so that it's not to give them a chance to get that resistance. So it's worth it to talk a little bit about resistance in that when we hear about resistance, often people are talking about superbugs. And so superbugs are organisms that have several genes for resistance to various classes of antibiotics. In some cases, it means they are actually resistant to different classes of antibiotics, which is kind of scary, really, when you think about it. And often when we talk about superbugs, the main one that we're talking about, the number one superbug, pretty much is number one on everybody's list, is this one here, MRSA. So this stands for methicillin-resistant Staphylococcus aureus. Although we're starting to see in the scientific literature, a lot of people, rather than methicillin, are using multi-drug resistance. So I'd mentioned way back, we were talking about penicillin. I mentioned that methicillin used to be the drug of last resort. So people would get staph infections, and they, the thought was, well, at least we have methicillin. We can treat them with that. So eventually, though, this is, I think, late 1980s, Staphylococcus started to evolve resistance to methicillin. And so methicillin-resistant Staphylococcus aureus is really kind of a scary name for a lot of practitioners. And so MRSA, sometimes people call it MRSA. I kind of don't like MRSA. It sort of sounds weird, so I always say MRSA, but you can use MRSA if you like. And you'll probably hear some people call it that. I find MRSA is more, I see it in the news as opposed to practitioners, but to each his own, I guess. It doesn't really make a big difference. It's still a scary organism. So let's talk a little bit about staph. We've kind of been talking about staph here and there, a little bit on and off. We're going to be talking about a lot more in the next few units and right to the end of the course, really. Today, if I was in person, you're probably wondering what this thing is on your notes. I was going to bring you my stuffed animals. I have a few stuffed microbes, and this one here is, of course, MRSA. And remember what Staphylococcus means? So Staphylococcus means you have clustered spheres, and you can see it's got some clustered spheres. Aureus is gold; it's kind of a golden color. And this one here is a superbug because it has a cape, and so you know, it's kind of a cute little organism. And I have a few others I was going to share with you. I see there's a question here. It says, do you blame the person who maybe had the staph infection who did not complete the full course of medication? I don't really know where to put the blame. You know, that's certainly also a very complicated question. The whole idea is, you know, everyone doing their part in terms of responsibly using antibiotics. And, you know, the more people that use them responsibly, the idea is you know, the more effective they're going to be for everybody. But certainly there is evidence to suggest that this can be a problem. You know, you can imagine there are elderly people, for example, that, you know, sometimes they have lung issues, and, you know, they get pneumonia quite frequently. Maybe every 2 to 3 years they're hospitalized, you know, let's say in their 60s or 70s. And eventually it gets harder to treat. And this is part of the problem, right? So anyway, I could spend a lot of time on that, but that's kind of the general practice, like I said, is to get people to make sure they take the full course to try to prevent the selection for resistance. So let's talk a little bit more about Staphylococcus. MRSA, I think I'd mentioned that Staphylococcus aureus is found on about a third to a quarter of us. So, you know, we have, I think, something like 64 students in our class, right? So probably, you know, maybe about 30 of us have Staphylococcus aureus on their skin. It's normally part of our, it's kind of a transient flora. It comes and goes. It's a natural thing. It's not really a big deal. But about a quarter of those people have drug-resistant MRSA. So if there were 64 students and a third of us, or did I say 30? I can't do my math. A third of 60 is 20. So a quarter of 20 is 5. So that means about 5 people in our class, maybe myself included, have MRSA on us. And if it's on the skin, it's not usually a big deal. The big deal is where it starts getting in other tissues. So we can have a lot of skin infections, so skin boils and rashes and things like that. And we're gonna talk more about MRSA later on. Skin infections tend to be not quite as serious, although they can, you know, obviously really suck. But getting into other tissues can be very, very serious. It turns out Staphylococcus is very good at infecting almost every single body system. And this is where it's scary. You can imagine people who are immunocompromised getting an MRSA infection can be very serious. So you can see I have some infections listed there, and many, many more. And the other thing about Staphylococcus, it's found on our skin. So that means in the environment, it can be found everywhere, right? Everywhere that people have touched, right? So doorknobs, clothing, it's been found on pets, it's been found on farm animals. So this is kind of scary. In some ways, it's a worse nightmare. It's relatively stable on those things. It can survive a little bit of drying up, unfortunately. Biologically, this thing is a nightmare. It has all sorts of enzymes and toxins, and like I said, is very good at infecting pretty much every single body system if it has the chance. Some other stats for you. In Canada, we're looking at 30,000+ patients a year and a couple of thousand deaths. Lots of costs, and you can see that last stat. So there's sort of a statistic out there in terms of people who stay in hospitals, and kind of depends on the hospital and the capacity. As hospitals get more full, the stats make it worse. But about 1 in 10 people in a hospital will get a secondary infection, and then if it's MRSA, that's an average stay of about 10 days longer. So that's very serious. It's not just a burden for people, but obviously hospitals as well in terms of cost and resources. Very, very serious. So I think I mentioned before, when you have these kind of patients, often they're isolated, and maybe special protocols such as getting visitors to wear gowns and gloves, wash their hands, and those kind of things. And the actual protocols vary from hospital to hospital, although I think there is some movement to try to get it a little bit more standardized. So by the way, there are a whole bunch of other bugs out there, and they often have acronyms. You can see VRE stands for vancomycin resistant enterococcus, or ESBL, or CRE, and so on. The one that we're going to talk about a little bit in the next few lectures is this one here, Clostridium difficile. I already mentioned that one last day, is something that forms endospores, and because there's endospores and has that stage where it's not growing, it tends to be more resistant to the drugs, and arguably that's the number 2 superbug. Pretty much a lot of the organisms that we've talked about, Neisseria gonorrhea, Streptococcus pneumonia, Pseudomonas aeruginosa, I can see I spelt pneumonia wrong, but that one has an E on the end for the organism name. A lot of them, there have been superbugs that are a lot of strains of these organisms that would be classified as superbugs. But the main one to think about is MRSA, and like I said, number 2, we can think about Clostridium difficile as kind of the number 2 superbug. There's a much longer list. Like I said, a whole bunch of these have been characterized, and we're always trying to control, and we're always concerned about these things. Okay, so you can see there's a couple of questions. Yeah, somebody's asking, should we know all these superbugs? Not really. We're mostly going to be talking about Clostridium over the next few lectures. We're going to be talking about Pseudomonas in topic 14. I know I've mentioned it a few times, and we're going to kind of just talk about a little bit more in topic 14. Streptococcus pneumonia, most strains are not considered superbugs, but just know that a lot of the organisms we've talked about, there are superbug versions of them. So what else can we do about these organisms? And usually every semester I get, 1 or 2 people ask me about this, because it's kind of something that's been in the news literally for decades now. Are there other things we can do to kill organisms besides just using drugs? Yes. What about using viruses? Yeah, actually, this is something that is viable. There are a few companies working on this kind of thing. And if you were living in the Soviet Union way back in the 60s, 70s, and 80s, there was actually a lot of active research using something called phage therapy. Remember, bacteriophage or viruses that infect bacteria with a certain amount of success. You can read about it there, and there's actually another really interesting article I was reading about. It was about, I can't remember. This woman's husband got ill. I can't remember what the infection was, but it was quite serious. And she was a microbiologist and spent some time kind of reading up on this. And they went from specialist to specialist, and he was just getting worse. And so as a kind of a shot in the dark, they flew over to Georgia. So not the state in the United States, but the country over in Eastern Europe. And they went to a pharmacy, and for $2 they got this this thing to drink, and it was supposed to be full of bacteriophage. And anyway, whatever was in there, it cleared up his infection. It was some sort of digestive infection, and probably saved his life. So we're kind of at the stage where people are taking this a lot more seriously. It's just really hard to research this in terms of what's going on in Canada and the United States. When you make drugs, the government wants to know what's in there. And so these virus things, we don't know what's in there. And that's kind of a problem. You can't just be feeding people in Canada things that we don't know what's in there. Drug and food administrations and regulations are really, really tight in our countries. And so it's something, we'll see. Last I read, there was actually a spray that one company had developed to put on, I think it was cheese or lunch meats, that was effective against salmonella. So, give it some time. Maybe this will become a real thing. It's very cool science, and just kind of a little footnote for this topic. Something else worth mentioning is what is kind of coming through and has taken about 20 years are these antibiotic stewardship programs. And it's kind of hard to explain what these are without going into like a full lecture on this, but you can see this stat here. And then in Canada, a certain percentage, it used to be higher than 50%. I think we're down to 20 to 50% is what the recent numbers I saw. But basically, there are a lot of antibiotic prescriptions that are not necessary, or they're inappropriate. By inappropriate, I mean somebody has a viral infection and they're getting an antibacterial drug. That's not good. People shouldn't be taking the wrong drugs, and taking the wrong drugs can, in the long run, lead to more resistance. And so there's all these programs that, these antibiotic stewardship programs that are kind of like I said, a sort of a newer thing last 20 years. And what they are is programs to have physicians kind of, really do two things, right? You can see the two things are stopping bugs and saving drugs, right? So using less drugs and finding ways to kind of be more strategic about antibiotic use. And these are the kind of things where traditionally, let's say somebody had an eye infection, right? And so, there would be a standard treatment, maybe gentamicin drops or something like that. And this was the standard treatment. So you can imagine every patient going through a certain clinic with an eye infection would get gentamicin drops. And this would go on for, let's say, decades. Now you can imagine that eventually, something is going to happen there, in that the organisms are going to start, getting resistant, and the gentamicin isn't going to work anymore. And some drugs, or like I said, these organisms can evolve resistance much faster to it. So often what's done with these programs is there's, the administrators are keeping track of what is prescribed, and sometimes they're actually mandating, right? They're telling physicians, okay, between January and June, you can prescribe this for eye infections. And between July and December, we're going to rotate through some different drugs. So you don't give the organisms a chance to necessarily evolve resistance. And this is the kind of thing that 20 years ago was almost impossible to do. Telling senior physicians what they had to prescribe. It takes strong administration to do that kind of thing. But the culture is changing, and these things are getting mandated. And so this might be something that you experience in your rotations as a nurse. So I'm not going to test you on this, but just to say that there's a lot of administrative things that can be done to kind of control what's going on with the antibiotics. Okay, so where are we at with time here? Okay, this is good. We're a little bit ahead of schedule, which is good, because I was hoping to try to do a little bit of catching up in the next couple of lectures. Just looking at the time, this is about where I wanted to finish off. And next day, we're going to talk about antivirals. I do want to spend the last 15 minutes here talking about the midterm, which is next Tuesday. So we'll finish the rest of this topic here on Thursday, and then we'll have our midterm on Tuesday. So let me just switch PowerPoints here. Just give me a moment. Okay, where is it? There it is. Okay, so let's talk about midterm number 2. So we're 1 week away from midterm number 2. And so we want to talk about what you need to know for midterm number 2, and a few other things. Okay, so let's kind of review what we're looking at. So midterm 2 is on Tuesday. It's in the classroom on paper, so make sure you bring a good pen. I'm working on probably using some sort of Scantron or something. I'm still looking into the technology, so filling in one of those bubble sheet kind of things. And I'll go over that on Tuesday once I have that sorted out, because we have a new system for that, which I haven't actually looked at in a lot of detail yet. So just hang tight on that, and I'll let you know how that's going to work if we end up using that. I'll give you some instructions on Tuesday. So this is going to cover topics 7 to 10. It doesn't mean you can completely ignore topics 1 to 6. It's just sort of assumed that you've studied topics 1 to 6 for midterm 1, and that we can now comfortably talk about, like I said, at this stage, there should be nobody asking what a virus is versus a bacterium or anything like that. So topics 7 to 10, this is going to be the format. There's going to be some multiple choice true false. There is going to be 10 marks for matching. There's going to be some short answer, fill in the blank, and I'll show you some sample questions of all of these types through the slides here. I'm going to have one scenario involving disinfection and sterilization. Excuse me, just losing my voice. And one case study. So that'll be 40 marks. So what did we cover? We covered culturing, identification, sterilization, disinfection, and our last topic that we're most of the way through is antimicrobials. So let's take a look at each of these in a little detail. Excuse me, I just have a foggy throat here. Might have to break for a glass of water in a second if I can't get my voice back. So microbial culturing. We talked about a bunch of things. We talked about things like media, what that is, requirements for growth. So things like temperature and pH and food for the microorganisms. We talked a little bit about methods for culturing viruses. And then we talked about some different media types. So the types of media that you really need to know are McConkey agar and blood agar. So remember, McConkey agar selects for gram negatives, and blood agar is a good way to distinguish between the streptococcus organisms. So the pathogens that were kind of covered a little bit in this unit, you can see there on the right. So what I've done is tried to kind of make a list of the pathogens that were featured. It's not a complete list, but it's the ones that were talked about the most in the unit. So just excuse me for a second. I am going to get a glass of water. I'll be right back. Okay, sorry about that. All right, so here's a sample true and false. An obligate anaerobe is a species that requires oxygen to live. Hopefully that's an easy one. The answer for that is, of course, false. Anaerobic means no oxygen. So make sure you read the question carefully and get the right answer. Second one, Helicobacter pylori is a gram-negative spiral-shaped bacterium that can live in the acidic environment of the stomach. So the answer for that is true. And that's kind of one of the main things to know about Helicobacter. It's one of the few organisms that kind of resides in the stomach as opposed to the intestines, where most of the bacteria like to live. Some of them can pass through the stomach, most of them don't live there. Helicobacter can live there. Remember, Helicobacter is associated with gastric ulcers and in some people. And but in a lot of us, maybe 50% of us actually have it as part of our normal flora. So it's not always a dangerous organism to have. Fill in the blank short answer. So it'll be just like the previous one. Some of them will be direct fill in the blank, like that first one. So you can see it says here only, and in that case, I'm actually giving you the words. Gram-positive or gram-negative bacteria can grow on McConkey agar. So the correct answer is gram-negative. And some of them will be, either definition-based, or they'll ask you for examples. If you remember, I'm just trying to think of the last one, midterm, I asked you for examples of protist parasites that were naturally occurring in Alberta. And I asked for 2 of them or something like that. This one here is a bit more definition-based. You can see selective media is, so what is it? So you're going to tell me what it is. It's media that allows one bacteria to grow and not another. And if you're answering things, just always a tip, examples are always a great thing to add. So in this case here, an example of selected media is McConkey agar, and McConkey agar, in this case, only gram-negatives grow. Gram-negatives grow. There we go. And I mean, if you want to give me more information about what's in the McConkey agar and all that, that's fine. Kind of depends on the question. But examples are always helpful. Even if it doesn't ask for examples, give them. And if you know them, it's only going to help you out. All right, topic 8 was microbial identification. And so we talked a lot about microscopy in topic 8. So, maybe it's good to know some of the ways we can get better images. And we really focused on gram stains. So if you remember, we made a giant flow chart of gram-negative and gram-positive organisms. So it's worth it to know a little bit about that one. I'm just trying to remember what I've had on previous midterms. I think, if I remember correctly, I may have had a matching question last year. I really can't recall. I know I've definitely had some that were multiple choice that said, which one of these is most likely to be, and so it could, let's say Staphylococcus. And so in case of Staphylococcus, you're looking at, the clustered spheres, right? And the tests will be in black and white. So if you're colorblind, there's no need to worry about this, but you should know the shapes and the organization of those organisms that were in the flow charts. We talked about acid-fast stains. We talked about some newer methods like fluorescence, serological testing, so using things like immunofluorescence or agglutination. We talked about ELISA and rapid tests, and we talked about the PCR or nucleic acid amplification tests. And so this topic actually covered a whole bunch of bacteria, some that were kind of tagged as special in here were the Mycobacterium tuberculosis, which is of course associated with the acid-fast test. And Neisseria meningitis was part of the case study, which is why I put it down here on the list. All right, let's take a look here. I got some sample multiple choice questions. So which of the following techniques could be used to visualize a live protist from a stool sample? So a live, living protist, okay? So here you go. When you go through multiple choice, always get rid of things that don't make sense. So if you remember, ELISA was not a method for visualizing anything. So we can cross that one off. Another thing is that electron microscopy kills the specimen. It's coated with heavy metal, usually it's dried out and all that, right? And so we're left with two selections, light microscopy and light microscopy with the acid-fast stain. So remember, the acid-fast stain is used for tuberculosis, not usually used for protists, and you've got this acid in there that's going to kill it. So the correct answer is A, light microscopy. Okay, so go through these logically. If you know what each of the things are, hopefully that helps you to answer the question. Here's another one. It says, which of the following could be used to analyze a sputum sample to detect for Mycobacterium tuberculosis? Okay. Okay, so sputum sample is basically what somebody has coughed up. And it says here, which of these could be used to detect for tuberculosis? So we talked about the acid-fast stain as kind of a traditional method for looking for tuberculosis. I also mentioned that there are modern fluorescent methods. And these methods here can also be used to detect tuberculosis. So the correct answer is all of the above. Just to clarify, I've been trying to not have any multiple choices with all of the above or any of the above, or both A and B, B, C, and D, but not E. I hate those questions. So this is an older one that I forgot to take out, but the answer is all the above. Like I said, I really don't like those questions, and they can be confusing. So I've been trying to eliminate them in general. Here's another one. It says here, okay, what if a technician left the crystal violet out of a gram stain procedure? Okay, so let's read the answers here. So remember, violet is purple. Okay, so anything with the answer purple is going to be wrong, because nothing can be purple if you haven't put the purple in. So that means all the cells will be purple. Wrong. Gram-positive, okay, that one is purple. That one is purple. Okay, so that leaves C and E. So let's read them. All cells will be pink, gram-positive cells will be colors, and gram-negative cells will be pink. So you have to go back to your gram stain procedure and remember what happens. And it turns out that the pink stain, the safranin, actually colors everything. You just don't see it on the gram-positives because the gram-positives are dark purple. So the correct answer is all cells. Okay, so what else do we have? We talked about sterilization and disinfection, and here's some definitions for you in terms of what is going on, right? We had sterilization, which is the killing of everything, and usually the one of the main methods for that is pressurized steam, sometimes ethylene oxide gas, and in the case of plastics, sometimes people are using UV sterilization, which is not, I wouldn't necessarily use the word sterilization, but it's pretty close anyway. Disinfection, we're often looking at using chemical disinfectants. And so there are a whole list of those kind of things, and you should go through and learn a little bit about all of them, and kind of, look at the important ones. So autoclaving, ethylene oxide, UV are important, and then look at some of the uses for surface disinfectants. Kind of the two main things are disinfecting, let's say, a bench top or something like that, and then of course disinfecting the skin. So those are two things to kind of think about while you're going through them. This is just a little flow chart I found in the textbook that's kind of trying to separate out and talk about all the different methods. So if you find these kind of flow chart things useful, then take a look at this slide. Certainly some of the things here are more useful than others. So sample fill in the blank, an autoclave. Most important thing to know is it's an instrument, and what does it use? It uses pressurized steam. So in this case, if you just use steam, I wouldn't give you the full mark, okay? It's important that it is pressurized. It actually makes the steam a lot hotter, or effectively hotter. True or false, endospores are resistant to autoclaving. The correct answer is false. That's why we use autoclaves. We're trying to kill those nasty endospores. Multiple choice, which of the following is an example of degerming? So you got to know your definitions. What was degerming again? It means kind of just physically removing the germs. And so B is the correct answer. And that's kind of the main thing when we're dealing with degerming, is we're scrubbing something away, whether it's scrubbing a patch of skin before getting an injection, or washing hands. Those are kind of the biggest examples of degerming. So sample case studies. Remember, I was going to give you that should say sample disinfection sterilization scenario. So I'm going to give you a scenario. This is one I've given in the past, and you're going to have to explain to me what you're going to do in this situation. And you can see in this example question is asking for 3 different methods to deal with the vomit on the floor, and asking you for a little bit of information about how the methods actually work to kill or eliminate microbes. So this will be one of your written questions. All right, so last topic that we're going to cover, we're just going to finish it on Thursday, is of course the antimicrobials. And so make sure you know some of the concepts around selective toxicity, things like how we get resistance, those kind of things. There's some things we haven't talked about yet in terms of what some criteria for selecting an antibiotic might be and whatnot, which we're going to kind of address a little bit at the end. And we have some pathogens, and so today we've talked about the bacteria. Next day we're going to talk about some of these viruses and other pathogens, and some of the treatment methods for them. And there's one drug for SARS-CoV-2 that you'll need to know as well, which we'll talk about next day, called remdesivir. There's our little picture of a bunch of the drug targets. So you can cross some of these off, or I'll circle them, right? So we've got penicillins, isoniazid, polymyxins. We have macrolides and tetracyclines. Macrolides that's erythromycin. And we have the sulfonamides and the fluoroquinolones, of which Cipro is kind of one of the more importantly prescribed ones of recent. So there's a picture talking about antiviral drug strategy. So that will be the topic of Thursday's lecture, at least part of it. and a bunch of other things that we will be covering. So things like, well, I guess we talked about testing effectiveness, considerations for use, resistant superbugs. So a lot of those things we have covered. Like I said, we're kind of 80% of the way through that topic. So probably it'll be about half hour of lecture that will cover the rest of these things, antivirals, antifungals, and so on. So sample short answer. This is probably the most important question of the entire course. Why should we not give penicillin to someone with a viral infection? This is the kind of question that, you know, if people get this wrong, I question whether I should pass them in the course. And that, you know, that's kind of a joke, but it's not a joke. Super important concept. Why do we not give penicillin for people who have the flu? Because penicillin is an antibacterial drug and the flu is for a virus. So make sure you can answer this question in some form. It pretty much guaranteed to be on this test or on the final exam somewhere. So make sure you can answer that. And sorry for going a couple minutes over time, but that's kind of it. So good luck on the midterm. I will take more questions about the midterm on Thursday, and you can contact me over email or visit my office hours at any time. Thanks for coming out. I will see you on Thursday.